Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today I will discuss a very important topic with you people, and the name of the topic is gram staining and the mechanism of action of on the cell wall. The dyes that we use in gram staining, what are basically their mechanism of action on the cell wall? Okay. I think you people are familiar with gram staining, but you know that why we perform gram staining to to differentiate the between gram positive and gram negative bacteria. Okay, the gram positive bacteria stains purple after the gram staining, and the gram negative stains pinkish color. Okay, so this video is basically about the concept that why this gram positive bacteria stains purple and why gram negative bacteria stains pink. Okay, so now first of all I will just discuss that what really gram staining is. What is gram staining, what is the procedure, in a minute or two. Okay, after that the main focus of my video is that what is basically the mechanism of action of the dyes that are used in the gram staining on the cell wall of these bacteria. Okay, what really happens to the cell walls of these bacteria that gram positive stains purple and gram negative stains pinkish. Okay, so stay tuned with the video, watch the video till end, and I guarantee that you will find this video very much important and it will clear up your concepts. Okay, so don't judge the duration of the video, because I will explain this in a very simple and in a very detailed way. Okay, so stay tuned and watch the video till the end. Okay, okay, let's start with the lecture. First of all, the procedure of gram staining. I think you will be knowing it, that what is the procedure. This is a very common procedure of the gram staining. I have taken it from the Google. So first, what we do, we take a colony, we make a smear on glass, this glass slide. After that, after making the smear, and so first what we do, we add crystal violet dye to the glass slide. Okay, and after that it retain it for 1 minute. After that we wash it with the water, the tap water. Okay, but keep this thing in mind that we should wash it with very gentle water. Okay, means that the pressure of the water will be not very much high, because it can also take, if it is high, so it can take away the colony with itself. Okay, so very gently you will rinse the glass slide with the water. After that you will add iodine for 1 minute, and after that you will rinse it with water. After that you will add alcohol, a few drops of alcohol for 10 seconds. Okay, and it is also known as decolorizer. Okay, and after that you will then add saffronin for 1 minute, and then you will rinse it with water, and after that you will dry it, and then you will look it into the microscope. Okay, so this is a very simple method. And at the last, in the microscope, the colonies that are purple in color will be known as gram positive, and the colonies that are pink in color will be known as gram negative. Okay, but my concern is that what happens when we add crystal violet, what happens when we add iodine, and then after that alcohol and saffronin. So when we add these components, what really happens inside the cell wall of that bacteria? Okay, if it is gram positive or gram negative, I am focusing on that. Okay, so let's start with that issue. Okay, so first of all, I want to tell you that I want to briefly illustrate that what really peptidoglycan is. Okay, we all know that our bacteria, if it is gram positive or gram negative, so there is a peptidoglycan layer. Okay, If it is gram positive bacteria, so there is a thick layer of peptidoglycan. If there is gram negative bacteria, there is a thin layer of peptidoglycan present inside its cell wall. Okay, so just look to the structure of peptidoglycan, then the molecular structure. So this is the molecular structure, and after that, this is the main physical structure of the peptidoglycan. I want to show this structure to you people just because I want to show you the protein content in this peptidoglycan. Okay, means that if just look here, this is alanine, this is glutamic acid, and this is d-alanine, and this is diaminopamelic acid. Okay, so these all are amino acids. This alanine, glutamic acid, and this d-alanine, these all are glutamic acid. Sorry, amino acids. So and after that, when two chains of peptidoglycan are linked with each other, so these are linked with glycine peptide interbridge. Okay, it is also known as pentaglycine bridge. Okay, so just look here. I want to show you the protein content, the amino acid content in the peptidoglycan layer. Okay, there are very high amount of amino acid content in the peptidoglycan. Okay, so and this is the structure of the peptidoglycan. Just look here. These are the this orange and yellow color are the neg and NAM, N-acetyl glucosamine, N-acetyl muramic acid. Okay, and after that these are the amino acids, and this is the this portion is the glycine, the pentaglycine bridge. Okay, so I was showing you this image, this structure, just to show you that there is a high amount of protein content, amino acid content inside this peptidoglycan. Okay, and just look here, there are certain spaces. Okay, a good peptidoglycan layer will be having spaces inside it. Okay, that materials can diffuse across it. Simply when it is a healthy peptidoglycan layer, so it will be having certain spaces in between it, and many water molecules and other dye molecules can diffuse inside it. Okay, so just keep this thing in mind. So this was a simple image of peptidoglycan. Just keep this structure in mind. It will help you in the later video. I was just showing you the structure. Okay, now here come the gram positive bacteria, the cell wall of gram positive bacteria. Now I will show you that what is the effect of crystal violet and iodine and alcohol and the safranine on these gram positive bacteria cell wall. Okay, this is the mechanism. So just look here. I want to show you this that this is a uh, this is a gram positive bacteria cell wall, the structure of gram positive bacteria. Okay, so from here to here, just look to the cursor. From here to here, it is a thick peptidoglycan layer. Studies have shown that 50 sheets of peptidoglycan layer is present in the gram positive bacteria, and these are the teichoic acids. These are the teichoic acids, and mainly the charge on the teichoic acids are mainly, I think so, it is positive charge. Okay, it is sorry, it is negative charge. The charge on the teichoic acid is negative charge. Why I'm telling you this, you will now come to know in a minute. Okay, so this is a brief structure of the cell wall of gram positive bacteria. Okay, so what happens in the first, we add crystal violet. Okay, this is crystal violet. Just to show you the crystal violet, CV means crystal violet. Okay, so when we add crystal violet to the gram positive bacteria, we are taking gram positive only. Okay, I will then show you that what happens in the gram negative in the later video. Okay, just keep if we are having a colony of gram positive bacteria. So when we add crystal violet color for 1 minute. Okay, so what happens? This crystal violet move inside the diffuses inside the peptidoglycan layer. As I have told you that there are spaces. Just look here, the spaces. There are spaces that a crystal violet dye can diffuse inside the peptidoglycan layer. Okay, so it passes the peptidoglycan layer and enters this periplasmic space. Okay, it can also enters the plasma membrane. Okay, it can also enter the plasma membrane easily, but just keep this thing in mind that it diffuses across the peptidoglycan layer. After that, What happens after 1 minute? We wash the slide, okay, after we rinse it with water. Okay, so what is the effect of the water on the peptidoglycan layer? So I want to tell you that the water can pass the peptidoglycan layer, but it cannot react with this crystal violet color, this crystal violet dye. Okay, water cannot react with crystal violet dye. Okay, so if we rinse it with water, so there is no such effect on the peptidoglycan layer on the cell wall of the bacteria. Okay, so after that, so the second step is that we add iodine. This is iodine. Okay, just I'm writing I. Okay, this is iodine. So what happens when we add iodine? So it acts as a modern and it binds with the, when it diffuses across the peptidoglycan layer, so it binds with the crystal violet dye. Okay, so after binding with crystal violet dye, so it stabilizes the crystal violet dye. Okay, so after that we rinse it with the water. I want to tell you a very important thing that mainly the crystal violet dye is basically it when we add crystal violet dye, so it converts into two parts. Okay, it converts into two parts. Just if I want to show it here in another color, if it is suppose this is a crystal violet dye. Okay, so I'm not going to the molecular structure, but keep this in mind. When we add crystal violet dye, so it converts into two components. Okay, the for one component is negatively charged and the other is the positively charged. Okay, the major component is positively charged and the other component is negatively charged. What happens here that the positive component, the main the dye component is basically positively charged. So this positively charged can then can also bind to this teichoic acid. Okay, and teichoic acids are only present inside the cell wall of the gram positive bacteria. So these crystal violet dye can also bind with the, if I want to show you here, it can also bind with the teichoic acid. Okay, so here it can also bind with the teichoic acid. Just this rectangle is the uh, basically the crystal violet dye. So also keep this thing in mind that it is positively charged, so it can bind to the teichoic acids. Okay, I was just telling you this. After that we add the iodine. So iodine can also iodine adds to the binds to the crystal violet dye. Okay, because iodine is a first of all, when we add crystal violet dye, so basically I want to tell you that one component is the dye component, the other is the chlorine component. Basically the molecular structure of crystal if you see it in the Google, so here is a larger structure and it is added to the chlorine part. Okay, so after that when the chlorine is removed, so iodine can easily bind to this crystal violet dye. Okay, it was just an addition, a detail one addition. Okay, so after that crystal violet dye can enter the diffuse through the peptidoglycan layer, and also it can even binds to the teichoic acid. Okay, after that we add iodine and then we rinse it in water, and at the last then we add the decolorizer, the alcohol. And keep this thing in mind, this is a very important step, the addition of decolorizer. Okay, and what happens that what basically a decolorizer is, so it is basically 95% alcohol. Okay, keep this thing in mind, the decolorizer that we are adding to the in our steps, so it is 95% alcohol. This means that it is very much concentrated alcohol. So what really is the effect of alcohol on the proteins, the amino acids of the peptidoglycan layer? So now it will help you there. If you have remembered the structure of the peptidoglycan layer, so it consists of a high amount of protein content. Okay, so what really happen when we add 95% alcohol to a few drops of alcohol to the peptide to our slide. So what happens that the alcohol, what does it do? It reacts with the it mainly reacts with the proteins and the amino acids of the peptidoglycan layer. Okay, so I want to show you, I want to change the color so it will then be clear to you. If we add alcohol, for example, this is alcohol, okay, and we add it into the peptidoglycan layer, okay. So rather diffusing diffusing across the peptidoglycan layer, what really happens when it is diffusing across the peptidoglycan layer? So because of its high concentration, it is continuously coagulating the proteins and denaturing the protein. I want to tell you that concentrated alcohol can coagulate proteins. Okay, it can denature the protein. It can disrupt the structures of the protein. Okay, the molecular structure of the protein. So what happens when we add alcohol to the concentrated alcohol to the cell wall of the this bacteria? So what really happens? Rather diffusing across the peptidoglycan layer, it denatures the proteins and the amino acid. So before reaching to the end of this structure, in the middle of the way, it is trapped inside the it is trapped in the it is trapped in the middle of the way, and this trapping is just because of that it is disrupting the and it is damaging the amino acids and the protein, so it shrinks the wall. Okay, it shrinks the peptidoglycan layer. So what really happen when it is diffusing? So it is coagulating the proteins. Okay, so what really happen, these spaces are then because of the denaturation of the proteins and the amino acid, these spaces are blocked. Okay, and the peptidoglycan layer shrinks. So this alcohol cannot get access to the cannot get access to the dye, the crystal violet dye. And after that, when we rinse it with water, so this alcohol can then easily be removed from this, so the dye remains. Okay, the crystal violet dye remains. So this is the reason that and after that, in the last step, when we add safranin, so that safranin can also not pass through the peptidoglycan layer, because for example, if I change the color for safranin, and it is, for example, this is the safranin. Okay, I want to show you, this is the safranin. So then when we add safranin, star is the safranin, so it cannot diffuse across the peptidoglycan layer, because it is shrinked and it is we can say the shape is disrupted, and the free spaces are then acquired by the denatured proteins. Okay, so there are no free spaces inside it, so it cannot diffuse. Okay, keep this thing in mind, it cannot diffuse. So after rinsing it with water, the safranin dye also evades away. Okay, so the crystal violet dye remains, and the our gram positive bacteria shows that it is the crystal violet dye remains, and it simply means that this is gram positive bacteria. And why it remains, I have told you guys. Okay, so this is basically the mechanism of the mechanism of the dyes that are acting on the gram positive bacteria. Hope you have understood it. Okay, now here we come for the gram negative bacteria. Okay, gram negative bacteria. So now here is the cell wall of gram negative bacteria. Now you will understand it well. Okay, first we add the crystal violet dye. Okay, first we add the crystal violet dye. So keep this thing in mind that it can diffuse across the plasma membrane. Okay, this is basically the outer membrane of the gram negative bacteria. Okay, so this is the cell wall of gram negative bacteria. So it diffuses the plasma membrane. After that, it easily diffuses the peptidoglycan layer. And keep this thing in mind that the peptidoglycan layer is very much thin in size. Okay, basically in gram positive bacteria it is of 50 sheets, but in gram negative bacteria it is of one or two or five sheets, okay, very much thin in size. So this crystal violet dye can easily enters the outer membrane, then it can easily diffuse to the across the peptidoglycan layer, and here is the crystal violet dye. After that, we rinse it with water, and we all know that water cannot react with the dye. Okay. After that, what happens that we add iodine, and same as the case, this iodine then binds with the crystal violet dye. Okay. Now here comes the main part, the alcohol part, okay, the decolorizing part. So what really happen when we add, if I want to show you there, for example, this is a decolorizer, okay, and when we add this decolorizer, and I have told you in earlier that it is 95% alcohol, very much concentrated alcohol. So what really happens when we add alcohol, we apply alcohol to the gram negative, the cell wall of the gram negative bacteria. So what happens that it can easily enters the outer membrane of the gram positive gram negative bacteria, and it can easily diffuses across the peptidoglycan layer, because here the peptidoglycan layer is very much thin in size. So before it coagulates the protein, it can easily enter the periplasmic space, okay, because the structure of the peptidoglycan layer is very much thin. So before it is trapped inside the peptidoglycan layer, and it can easily enters the periplasmic space. So after that, what happens, this alcohol, what does this alcohol do? It reacts with the crystal violet dye, okay. This alcohol reacts with the crystal violet dye, and it remove it decolorizes the crystal violet dye. Okay, I will show you the chemical reaction that how it decolorizes the crystal violet dye, but just for now, just keep this thing in mind. So this alcohol reacts with the crystal violet dye, and it decolorize it, okay. And after that, if we rinse it with water, so that water can then easily remove this crystal violet dye, or if it does not remove it, but keep this thing in mind that the alcohol has made it has done its work. And what is its work? That it decolorizes the dye. And after that, when we add saffronin, so then that saffronin can easily pass through the outer membrane and enters diffuses across the peptidoglycan layer, and it can accumulate here, okay. It can accumulate here, and we all know that by adding saffronin is purple in color, so the gram negative bacteria show purple color, okay. So this was all about the gram negative bacteria. If I want to show you that this is the chemical reaction, that what do the alcohol do with the crystal violet dye. So this is the structure of crystal violet dye, okay. This is the chemical structure of crystal violet dye. And for example, this is, for example, we say ethyl alcohol, okay. So this is CH3OH. So mainly the OH part, this OH part of the alcohol reacts with the crystal violet dye, and it this change its structure, and it remove the dye part, okay. It removed the dye part. So just look here, the crystal violet has lose its dye part, and now it is decolorized by the alcohol, okay. So this is just it get access to the crystal violet dye and decolorize it in the gram negative bacteria, while in gram positive bacteria it cannot get access to crystal violet dye, because it is entrapped inside the thick layer of the peptidoglycan, okay. So just keep this thing in mind. If I want to show you a very the last slide is that this is very important steps, and keep this thing in mind, that crystal violet dye, this is a crystal violet dye that is binded with iodine, the more mordant, okay. So crystal violet is insoluble in H2O. I told you in the first in the earlier part of this video that H2O have no concern with the crystal violet dye, and it cannot react with it, it cannot solubilize with the crystal violet dye, okay. After that, just look here, alcohol dehydrate gram positive bacteria, means that it what does it do? It shrinks the peptidoglycan layer, okay. and crystal violet dye gets trapped and retain washing. Okay, this was the step I was telling you. And in gram negative bacteria, what happens? Is easily decolorized by the alcohol, and this is just because that the peptidoglycan layer is very much thin in size. Okay, so this was all about the mechanism of gram staining. That we all know that what really gram staining is and what is the procedure. First add the crystal violet dye, and then the iodine, then the decolorizer, and then the saffron. It is known by every person, but mainly as a microbiologist, a person should know that what really happens to the cell wall of the bacteria, why gram positive stains purple and why gram negative stains pink. Okay, so this was all about the video. If you have any further questions, you can ask me in the comment box. I will also answer you there. Thank you very much for your concentration. If you have understand this video well, just like this video, add your comments and subscribe to my channel so that I should be more motivated and make many videos related to science and microbiology. Okay, thank you very much. God bless you.